Yeah, Genebank CRP is the 16th CRP, and in this presentation I will take you along how was the development of this CRP, and more importantly, what are the components in terms of objectives, activities, wherein not only the main gene bank but all the three regional gene bank of ICRISAT are involved. I acknowledge in beginning itself the contributions of the gene bank managers from Niamey, Hamidou Falalo, and earlier Tom Hess, from Nairobi Mary Mugonja and Gangarao, and from Bulbayu Sakile. Beside two regional directors in West and Central Africa and Eastern and Southern Africa, Dr. Farid Baliar and Dr. Said Slim. And this gene bank is really, Genebank CRP is in fact a tripartite partnership between the all 11 CG centers which do have gene bank. And ICRAF was the new entrant because it was initially not involved in that. Global Crop Diversity Trust and CGIR. And this is really a plan of 5 years, 2012 to 2016. And this plan envisages the management of global collections in 11 CG gene bank, which is about more than 700,000 accessions of various species. And not only that, but to secure the sustainable funding for these collections in the 11 gene banks. And what this CRP will accomplish is that it will fulfill its global obligation of providing germplasm as an international public good, meeting international standards. And this is a very critical because several times whenever these international scientific standards in terms of gene bank has been lowered down, it has created a lot of problem. A recent example is Griffin Gene Bank, which does have the Arachis germplasm in USA. Viability of some of those accessions is not more than 30 to 40%. Whereas at international standards, including our regional gene banks, we do envisage a viability of not less than 85%. So those rigorous international scientific standards need to be observed. Also, it ensures accountability and enhancing cost efficiency and long-term sustainability of these collections. And it comes through nurturing collaborations and enhancing communication. And how these collections are going to be managed, because CG gene banks, as I made a mention, do have a very large collection, which is about 10% of total global collection. Global collections are about 7.2 millions and about 1800 gene bank, but these 11 gene bank themselves do have about 700,000 accessions. And they would be available according to legal requirement, and that is following a standard material transfer agreement of International Treaty on Plant Genetic Resources for Food and Agriculture, and promoting and development of a rational global system. This is a very long term, because in terms of rational global system, how that rationality is being achieved, particularly in case of when crops are common. Chickpea in case of Icarda and ICRISAT, about 9,000 accessions are common. Even within collections there are common, say duplicates and other things. And cost effectiveness, efficiency and sustained management of these to maximize efficiencies. Also, it need to provide benefits in terms of materials to the centers, CG centers, national programs, NGOs as well as private sectors. And The Genebank CRP, though, is the latest entrant as the 16th one, but the work really started long back in 2007-2009 when we were having GPG2, that is Global Public Goods Project 2 from the World Bank, and before that we have GPG1. And in one of the activity of Global Public Goods Project 2 was to estimate the cost of typical gene bank functions like conservation and others. I'll cover them in detail. And 3 gene banks, ICRISAT, CIMMIT and IRI, were chosen to have estimate of each of the activity accession-wise. And a consultant, Daniela Horn, spent about 2 weeks' time at ICRISAT Gene Bank. And ICRISAT Main Gene Bank and 3 regional gene banks were costed. Each activity accession-wise, crop-wise, accession-wise. And details, all details were collected, and that's how this process started in around 2008-2009. Gene bank costing study identified all costs of standard activities which are predictable and which are core to the functioning of the gene banks. Study also identified what kind of non-recurring items could be there, that if number of accessions increases, we may require a cold room. And there could be items which are multi-center by nature. Items which are inherently expandable, say evaluation of germplasm, molecular characterization of germplasm, they were also identified, but they were not included in the gene bank proposal. And for 2012, the budget was 21 million. Of that, the contribution from Global Crop Diversity Trust was about 10%, 2.1 million. And there are 4 objectives and 11 outputs of the Genebank CRP. And first objective is that crop and tree was added when we got a crop into this. And these collections are covered under Article 15 of the treaty, International Treaty of Plant Genetic Resources for Food and Agriculture. So under this objective, both long-term and medium-term storage. So you can estimate the cost at how long the accession could be viable. And we keep at -20 degrees Celsius, as you know, about 110,000 of our 120,000 accession. ICRISAT Gene Bank has at the moment about 120,000 accessions from 144 countries. 5 mandate crop, chickpea, pigeon pea, sorghum, pearl millet, and then 6 small millets. And we also have medium term, wherein 4 degrees Celsius and 30% relative humidity, all 120,000 accessions are conserved. Safety duplication was regarded as an important component to take care of situations like civil unrest or something like that is happening in Syria for the ICARDA Gene Bank. And at Svalbard, every accession of CG Gene Bank has to be duplicated. And by last year, October 2012, we have deposited 97,800 accessions from ICRISAT Gene Bank. So about 90% of our accessions has been deposited there. Objective 2, conserved germplasm is clean, thoroughly tested for diseases, and available for dissemination. And that's why whenever we get new germplasm, that is taken in all the cases under a standard material transfer agreement, so that there is no problem in making that available to anybody who require. Regeneration and characterization. All germplasm has to be regenerated, multiplied, and characterized. And characterized here is different than the evaluation which we typically take in case of plant breeding and other things. Here characterization means for very, very standard set of morpho-agronomic traits. Disease testing and cleaning. If there is any accession which does show any kind of disease where is problem, then it is cleaned and then conserved. Introduction of new accession, this was recognized almost at the rate of about 1% per year. So in 5 years, you get about 7 to 8% accession. So if we are having at the moment, at that time we were having 119,000. So it's envisaged that by 2000 or 2016, ICRISAT gene banks should have about 128,000. And within a year, we already have 1,000 accessions. So I think we are doing pretty well there. Also, beside that, Global Crop Diversity Trust has a project which is operating in 77 countries, and top 246 collections are being regenerated. And all those collections which are regenerated under this project has to be duplicated into CG gene banks. So our mandate crop as well as our small millets, germplasm, ICRISAT receives those germplasm from these countries. Then multiplication and dissemination. And dissemination, how you take that when costing is to be done, because this is a very variable character, very variable. Supply can be in 1 year 8,000, another year 2,000, and also several gene banks, particularly Africa rice and IITA gene bank in certain crops have very low dissemination rate. But in our case, during that period, which was taken into account 8, 9, 10, we had reasonable, almost on average supply. Because in last 37 years, we have provided about 1.4 million samples to scientists in 147 countries. And that's a very remarkable good number. Objective 3 was use of conserved crop and tree diversity. And this was basically through managing the information on accessions, and then Genesis, a new tool which would be replacing Singer, is in place, has to be in place. And objective 4, crop and tree diversity conserved for global rationalized system. And in this objective, the activities mainly are developing partnership and exchange of services, rationalizing and optimizing collections. So this will be taken at a much later stage because it's a quite sensitive issue. Establishing and updating quality management systems, operational manuals, and staff retention plan, because gene bank activities are specific activities and we need to have staff on board before the old staff retires. And this was recognized in the meetings, and a provision was made. And then ICRISAT gene bank does have manual, and eventually all the gene bank by 2016 may have to obtain an ISO. Now these are the activities which were considered very essential, vital, but not included because the budget could have gone overhead. So gap analysis and collecting. Collection are funded, but through special funding provisions. Somebody puts a proposal that he wants to do collecting in these 3-4 countries, then money is given. But as such, it was not costed into the gene bank funding. Molecular characterization, no, it wasn't. Development of core, mini-core collections or fixed subsets, and evaluation of germplasm for important traits. This was considered that some of these activities would be in the crop-specific CRPs and not the part of this. And that's why when grain, legumes, and dryland cereals were being developed, these activities were put over there. Pre-breeding and even training and technical backstopping. For that, additional funds are available for the gene bank managers. Other activities were, which were not included but recognized: research on conservation methodology, reproduction biology, and taxonomy. Because that's essential, because in several situations it becomes difficult to assign species to the particular collecting objects. Development and operation of Genesis, this was again kept separate, and network and providing international leadership and facilitation, public awareness, attendance to conference, and visitor service. This has again additional component than the Genebank funding. Partnership to achieve the objectives of Genebank CRP, Global Crop Diversity Trust, and then 11 CGIR Center work together. Svalbard Global Seed Vault as a safety measure of having all the collections from the CGIR centers and other gene banks. Secretariat of the Pacific Community, this was also recognized an important partner. United States Department of Agriculture, particularly for providing and harmonizing the standards of the gene bank and for safety duplication at their site. Non-CGR gene bank hosting safety duplicates of say in ICRISAT, pearl millet, germplasm is duplicated in Canada, and then research organization providing service for plant health and cryopreservation. Breeding program, both in CGIR and NARS, because they are the one who uses this germplasm. Research programs and others, national programs and organizations and NGOs. Then monitoring and evaluation work plan were developed. And for this, baseline data for each of the activity was developed. And then we have been developing the multi-year budget. On 22nd and 23rd, I had meeting in Nairobi with the gene bank managers and we are working on this. Then annual work plan and reporting, main activities and progress towards achieving those standards and what are the obstacles. Then annual meeting of the gene bank staff or gene bank managers, and then consolidating standards and strategies. And each center has to undergo testing, and then in 2014, ICRISAT gene bank would be reviewed. This is timeline, annual performance management and financial report previous years, 5-year review, development of cost saving proposals and arrangements. And these are the risks: inadequate funding, natural disasters, civil unrest, political factors. However, Genebank CRP strives target of 100% safety duplication of CGI gene bank at Global Seedbed Svalbard. And this is the management team. We have a representative from consortium. Unfortunately, we don't have representative from any of the CG centers. And thank you very much.
Thank you, Hari. Questions? Yeah, Hari.
In your seed health testing, you said looking for disease organisms, but I thought the major threat is from the storage pests, which are around 50 species. And pests like storage mites and nematodes, they can even survive even very low temperatures, around 10 degrees centigrade or below. So maybe this may be addressed, at least in ICRISAT gene bank.
These activities really were costed not having a particular gene bank in mind, but the activity which could be common across every gene bank. So there is no point in saying that somebody is costing something more than the others. So if there were some specific activity for a specific gene bank, for instance, cryopreservation for SIP, they got much more money for that activity. But cryopreservation was not recognized as a uniform for other gene bank, except for vegetatively propagated crops.
Mary?